For this lecture for Bio 412 Comparative Genomics, I'm going to be talking about prokaryotic genomes. Specifically, some of the learning outcomes associated with this lecture are that I would like for students to be able to recognize the three major divisions of cellular life, know the features that distinguish the divisions of life, including viruses and how they reflect differences in their genomes and structures, understand the molecular basis of adaptation, recognize the functional genomics underlying specific bacterial traits such as pathogenicity, and appreciate the developments that have been made in metagenomics. So first off, if we start talking about prokaryotes or virus genomes, we should define what a prokaryote is. So prokaryote is what's seen as a relatively simple, we'll put that in quotation, simple form of life in which cells lack a nucleus. Why do we care about prokaryotes? Well, for us, they're a major source, or one of the major sources, if not the major source, of etiological agents for infectious disease. In terms of being relatively simple, the fundamental principles, what we're referring to there is that the fundamental principles of metabolism, genetics, and gene regulation are all sort of a little bit more straightforward than what we see in eukaryotes. So they can be very helpful in that regard in terms of as models for us understanding these processes, as well as for use in things like biotechnology. They represent some of the earliest forms of life, being up to be about 3.8 billion years old or so. They are important mediators of ecological and geological cycles. This includes photosynthesis and being responsible for the rise of atmospheric O2. They are major carbon sources. If we look over here on this table on the right, we can see the various habitats that we see in the world, and the number of cells, and the number of the amount of mass of total carbon kept in prokaryotes. And what we can see is that there's about as much carbon kept in prokaryotic organisms as we find in almost all of plants, depending on how you do the calculations. And additionally, they're responsible for being also the repository for almost 10 times the nitrogen and phosphorus that we would find in plants as well. So we always talk about plants being a sink for those kinds of nutrients, but bacteria, other sorts of prokaryotes, are also incredibly important in that regard. And even if we just think about the human body, right, the ecology of the human body, there's about 100 trillion bacterial cells in the human body, and there's only 37 trillion cells that are human cells in our bodies. So they obviously can have a huge effect on our bodies and our health as well. And finally, prokaryotes and their processes are key components for a ton of various types of biotechnological advancement, not the least of which would be, the advent of systems like CRISPR-Cas9 and gene editing and all the things that come with that. So incredibly important systems. So it's important that we understand them and understand their genomes. There's basically a few major types of prokaryotes. Often we would break them up into the categories of Archaea and bacteria. One of the main difference here is the presence of introns, with Archaea having the capacity to have introns in their genes, whereas bacteria generally do not. Each of these two species have different types of tRNAs and DNA replication enzymes associated with each taxonomic group. Archaea actually have some histone-like DNA binding proteins, so they can actually do some things that look similar to what eukaryotes are doing in terms of binding DNA. And then if we look at their cellular membranes, we can see distinct differences between them, where the bacterial cellular membranes contain D-glycerol and fatty acids, whereas Archaea cellular membranes contain L-glycerol and polyisoprenes. And additionally, there are these linkages between these side chains of glycerol, which are ether linkages shown over here in Archaea, and are ester linkages here shown in bacteria. So very different structures in terms of how their cellular membranes are being maintained, the biochemistry of their cellular membranes, as well as we see some differences in their cell walls, with bacterial cell walls containing peptidoglycans. We also see some big differences in the metabolic pathways that are sort of that we might find in each of these two different groups, and we'll go into that in more detail. One way that we might think of how we are able to distinguish relationships between these big taxonomic groups is by finding what is the common homolog or ortholog that we can use. And in this case, what we would use would be a 16S ribosomal RNA, because all archaea and bacteria have one of these 16S rRNA genes. And we even find one in eukarya. In the eukarya, actually the homolog in the ribosomal subunits is actually an 18S sequence, but we do find a 16S sequence in the mitochondria. So we can use those as a way of looking for similarities between these two, right? And so you can see, just looking at the two-dimensional structures, there's a lot of similarity in the structure of this gene, of this RNA. We can see sort of the blue and the red part of it outlined up here, which shows some of the variable structures. And if we look across a whole bunch of different archaea and bacteria and eukarya, we can use a phylogenetic tree to sort of tease out what are the relationships between these three major groups of organisms, major divisions of cellular life, as it were. And this brings us to sort of a classic point of interest for biologists and evolutionary biologists, and that is to search for what is known as the last universal common ancestor. So in here we're trying to say, well, we've got the tree of life here, with bacteria, archaea, and eukarya. What's the relationship between these, and what did life look like when it began? Can we use our understanding of these three large divisions of life, of cellular life, to be able to draw some inference about that? And by looking at these type of 16S sequences, one of the things that we note is that archaea is actually more closely related to us than bacteria is. And what it also looks like is archaea is perhaps the most closely associated, existing archaea species could be the most closely associated with what would have been the last universal common ancestor for all life. So this is all based upon the work of Carl Woese. And one of the things that this relationship does show is that, because at the beginning we've got archaea down here in close relationship to this sort of common ancestor between archaea and bacteria, that there was a lot of horizontal gene transfer sort of early in this process. So there's a lot of gene exchange. And so you end up with a lot of shared genes between these two groups especially. It's important when we think about prokaryotes to think about how their genome is organized. Prokaryotic cellular structure is a lot different than eukaryotic cellular structure. The genome is located in a nucleoid, and so there are no membrane-based organelles like mitochondria or endoplasmic reticulum or anything like that, right? Basically, everything's just kind of loose and floating in the cell, and the genome is not packed into a nucleus. Instead, it just forms this nucleoid region. It can range anywhere between 600,000 base pairs to 10 million or 10 megabases. So that's about the size of when we look at these genomes. And, along with the nucleoid region, we have our cellular cytoplasmic membrane. We've got ribosomes and the capsule that surrounds it, as well as pili, flagella, and cilia that can use for the bacteria to move around, and some other items in their genome as well, like plasmids, right? So their genome, prokaryotic genome, is not just organized into this nucleoid region. They also have these short sequences that can exist within the cytoplasm. Typically, they're these short circular double-stranded DNA molecules. They can be anywhere between 100 to 1,000 base pairs to several million base pairs long. But these are also one of the ways that genetic information is stored. There is, within the genomes of prokaryotes, we do find evidence for some insertion of sequences so that you can get repetitive elements or mobile genetic elements infecting these genomes. But again, these have pretty small genome sizes, so it's not like a dominant feature of these genomes. And one of the things that we do note is that they generally lack introns and have very short intergenic regions. So one of the reasons why their genomes are so small is that they don't have a lot of repetitive element. They don't have a lot of quote-unquote junk DNA in their genomes. They're actually not able to tolerate that very well. If we think about a genome like E. coli, we can see that a classic model species, it's got a genome of about 4.6 megabases, and of that, there's about 4,500 genes. About 87% of them are going to be about coding sequence. There's some regulatory genes. There's some other RNA structural genes. Average gene size is pretty short. It's around 900 base pairs long. So we're not talking about really long proteins for the vast majority of these genes. When we think about how bacterial genomes are replicated and how they're transcribed, it shows us some of the idiosyncrasies that's important for the organization of this genome. So typically what happens is an organism like E. coli or any other organism will start off and it's got about 5,000 genes or so. It's going to have an origin of replication region, and then it's going to bidirectionally replicate from those in both directions, right? So when it does replication, it's going to start there and then go out from in either direction. There are genes on both the positive and the negative strain, as indicated here by these red and blue arrows. And one of the things that's typical about how bacterial genomes work is that they're organized into co-transcriptional units or operons. You guys should have learned about these, all the way back in IntroBio. I talked about the lac operon, and in IntroBiolab you would have done experiments looking at the arabinose. Using an arabinose operon, so that we could see, the role that basically you'll have a promoter and it'll activate a suite of genes that are under that one promoter. So it tends to express and translate several genes at the same time. This is an example of, a set of genes that are all under the His operon, for instance, right? So this here is an example of the His operon, and then a set of protein coding genes associated with it. One of the interesting things about bacteria and Archaea as well is that they have very different methods of DNA transfer than what we might expect, or what we associate with eukaryotes, right? So we tend to think of eukaryotic genetics being dominated by a vertical gene transfer model, whereas Archaea and bacteria have a lot of other methods for DNA transfer that allow for a lot of horizontal gene transfer. So not necessarily from parent to offspring, but just among co-occurring members of their community. So the way that they can do this is through things like transformation, where they can just uptake naked DNA that's floating around in the environment. They can do this through a process known as conjugation, where there's some sort of insertion of some or all the DNA from one cell to another. It's kind of like bacterial mating, and this is one of the processes that can allow for recombination to occur. And then there's also evidence that DNA can be transferred via a process known as transduction, where DNA from one cell is kind of sucked up, a section of it is sucked up by something like a bacteriophage, and then that virus then manages to transfer that DNA to another cell. Archaea differs from bacteria in a lot of ways, but one of the most sort of prevalent ways that we've identified these differences is that Archaea tend to be associated with a lot of extreme environments. So we call them extremophiles. So either high temperatures, high chemical gradients, lots of salt, right? And one of the questions for these organisms is whether or not we think that they originated from either a mesophilic or a thermophilic environment. Also important to note that a lot of Archaea are marine, so about 20% of them are marine. And why might this be important? I mean, we want to think about this in terms of that Luca argument that we were talking before about our last universal common ancestor. If we think Archaea might be closer to our ancient origins, maybe it's this extreme lifestyle, right, which is indicative of that evolutionary history, because initially the Earth would have been a lot less hospitable and might have been in a more extreme case, in a more extreme chemical state, in a more extreme temperature state, etc. If we look at the major divisions of Archaea, we can see that there are sort of some major branches here. We have the Eukaryota and the Cranarcharyota. And for the Cranarcharyota, we tend to find a lot more thermophiles on that side, whereas we tend to find like more of the methanogens, sulfur reducers, and acidophiles, halophiles, etc., over on the other side of the tree. What is interesting when you think about these Archaean organisms is how can they actually live in these extreme temperatures, and it tells us a lot about adaptation. So one of the key uses or ways that we look at these organisms is to understand sort of like what is the nature of being able to survive in an extreme environment, to adapt to it. And one example is people have looked at things like thermostability. So there's this idea that thermostability, especially in DNA, is going to increase as you increase the GC content of DNA. So there's this idea that perhaps the differences that we observe in GC content has to do with an evolutionary history in the organism's ability to adapt to specific extreme temperatures. So in 2003, Singer and Hickey looked at the genomes of 8 archaeal and mesophile thermophiles and 32 bacterial mesophile and thermophiles. And then they tried to see which genome characteristics correlated with growth temperature, optimal growth temperature. And one of the things that they found is that GC was not correlated with growth temperature. So it didn't actually look like the percent G. We know that chemically, the more the higher GC content your genome has in it, the more stable the DNA will be. But this was not necessarily mean that it's going to be more closely associated with the ideal growth temperature for these organisms. But instead, there were other things that helped establish DNA stability, like various ligands, DNA binding ligands. Those tended to track more better with the system. What we did see is that for RNA, for non-coding protein RNA and codon usage patterns, that these had GC content that was definitely correlated with growth patterns. So in sort of the less stable, more volatile nucleotide molecule rRNA, we did see the GC content was actually higher in those thermostressed organisms as opposed to non. And then we saw that specific codons tended to be favored. There was a GC bias where tended to have the codon favored when there were multiple synonymous codons, tend to favor ones that were ending in C and G, as they may provide greater thermostability to the coding sequence. Lastly, we saw a wide range of effects where, thermostressed organisms tended to have smaller proteins, large, but they had more of a charge. They had they also had more salt bridges in order to stabilize the thermostressed proteins. They had greater conformational freedom, so they had more flexibility in themselves. So they seemed to be able to function under more stressful situations. So there's a variety of other adaptations that we can see at a sequence level that would look to have that could indicate what are some of the ways that these organisms actually adapt to extreme temperatures. An interesting case for examining these types of things, to say we just wanted to look at one specific genome, we could look at something like Thermococcus cotocarensis. And in this study, in a study in the, I think, in 2005 study on this organism's genome, this is a sulfur spring thermophilic Archaean. It's a strict anaerobe that basically survives by reducing elemental sulfur to H2S. And as the researchers in this study, Foucault et al., basically went through and did the sequence of this 2 megabase genome. And in the process, started going through and identifying, genes that they found that they could associate with different functions. End up discovering about 261 genes which were specifically unique to Thermococcus cotocarensis, of which 15 were identified as intines, which can be used to which have an identity to be involved in basically alternative splicing in Archaeans. So there were some interesting components of their genome in that regard. What was also interesting to see was that this Thermococcus genome, it had about quite a few genes, these 261 genes that were unique to the organism that weren't easily identifiable from other resources. It also was missing about 4 different amino acid biosynthesis pipelines. So it didn't have the ability. Here in red, it kind of highlights amino acid systems that it's not able to build all that effectively. And so isoleucine, leucine, valine, arginine were just basically almost entirely lacking from this organism. So that's at least 4 amino acids that it won't be able to create itself. And it remained to be seen whether or not phenylalanine biosynthesis was well supported as well. So despite the fact that it's got all these processes, it's interesting to note that there are these limitations that are being put, and it actually can't create the entire set of amino acids that it would need to create all the proteins that all the amino acid set that you might expect to find in an organism. What's interesting in comparing this genome, so we think about these unique genes, and we compare this genome with its closest relatives, right? these other species of what are Pyrococcus, basically. Pyrococcus furiosus, Horikoshi, and Abyssei. And what we see here is we see genomes that are approximately the same size. Cotocorensis is a little bit bigger. We do see an increase in GC content. So perhaps this is indicative of the thermophilic nature of this organism. But I think what's really interesting in this case is if we look at, we see, in terms of general coding sequence, we see, similar processes. But if we look at the relationship between these closely related organisms, what we are able to identify are these large synthetic regions, right? So there are these, we can look at Abyssei and Horikoshi, and we can see that there looks to be like a large inversion here, where we have the genome which has been inverted and flipped. We've got areas of synteny. So in these closely related species, we also see that there are sort of these similar patterns between Horikoshi and Furiosus. But then when we looked at its relationship of Furiosus to Cotocorensis, it's pretty interesting to see is that like all the parts of Cotocorensis are actually, you can find almost all of them in Furiosus. So it's not like these genes don't exist, these 261, like almost all the genome is not unique to Cotocorensis. It's just that it's incredibly more mixed up. There's been a ton of either horizontal, recombination or rearrangement in this organism that has allowed it to basically form all sorts of unique genes and things of that sort. So it's actually not that there's like a lot of horizontal gene transfer putting new genes into Thermococcus, but it's actually just that there's been a ton of different rearrangement going on between these two organisms since they were diverged from each other. So pretty interesting story. So that tells us that, how the pattern of evolution it's not one that's dominated by horizontal gene transfer. It's actually one that's dominated in this case by a rearrangement leading to providing the genetic machine engine for adaptation. So an interesting case where we can take a multiple comparative perspective, and it tells us something really interesting about how adaptation might have occurred in this organism. What about bacteria? Well, there's a lot of reasons why we would be interested in bacteria. One of the principal reasons why we're often interested in bacteria, especially from a human perspective, is the role that they have in pathogenic disease. So when we talk about pathogenicity, basically it's the ability of an organism to cause a disease. And we often refer to the virulence of that disease as the degree of pathology caused by that organism. So the more virulent it is, the more extreme the pathology. The less virulent it is, the less extreme the pathology. When we think about what causes virulence, we usually identify that causative factor as something referred to as the virulence factor. And this is the component that leads to a pathology, whether it's a toxin, a surface protein, or a defense shield. What is one of the things that we've noticed a lot with pathogenicity as a trait is that for bacteria, one of the reasons why so many bacteria may be involved in pathogenic responses for us is that a lot of the pathogenicity genes, the things that underlie pathogenicity, tend to be able to be transferable via horizontal gene transfer. So this leads us to a question about, when we have these horizontal gene transfer processes, especially in organisms like bacteria, is whether or not they are really actually separate genomes or it's all just sort of one genome of the community. There is an active debate going on in biology about, the nature of genetic transfer in these organisms and whether it's dominated more by the environment, and that is sort of the thing that establishes the sets of genomes that are available to organisms, or if it's more determined by vertical processes where genes are still being inherited, and horizontal gene transfer is more of just complementary piece to that. But it brings up a lot of really interesting questions about when organisms start changing genes back and forth. At what point do we actually call them species? Or should we just consider all bacterial organisms to have sort of one universal genome? It's a fun little intellectual argument. But let's look at some examples about like what might lead to something like pathogenicity, and we can see that taking a genomics perspective on this can be very informative. So if we compare something like two different strains of E. coli, so if we look at a K12 strain and we look at a 0157 H7 strain. And so if we look at these two different strains, we can see, the K12 is pathogenic, where the other causes hemorrhagic colloides. We can see a difference in their genome sizes, right? Like the 0157 strain is almost the 0157 H7 strain is about almost 1 million bases larger. And we can see if we compare them that there's about 1.4 megabases shared between the two genomes, and there's large regions that are unshared between them. And the regions that are unshared tend to be genes that cluster together. And so if we look at the strain-specific genes in each of these regions, we can see that there's, about 1,300 of them that are specific in the K12 strain, whereas in the pathogenic strain, there's about 528 genes in that region. So, and one thing that we note about that region of these unshared genes is that they have a very different base composition. In other words, GC percentage, what is the percent genome, what is the percent base makeup? And so what that tells us typically is when we see a segment of a genome which has a very different base percentage, is that region may have come from another organism, because generally organisms will have a pretty consistent percent GC across the whole genome. They wouldn't change dramatically across the genome. And so when we see these big differences, this might indicate to us that there's been some sort of horizontal gene transfer in from something that is a very different organism. One interesting case to think about pathogenicity is the genome of Staphylococcus aureus. So if we think about this staph, I think we've all probably heard at some point of MRSA, or methicillin-resistant Staphylococcus aureus, right? And one of the things that you can do is you can look at different strains of MRSA, and these are all regions. RD on the y-axis indicates regions of differences between different strains. So there's about 18 different regions that are different among various strains of Staphylococcus aureus. And wherever you see it hatched, right, those are examples of differences that can be differential regions that are different that can be associated with those hatched ones, or strains that are actually MRSA strains, so they're methicillin resistant. And the red ones are ones that can be associated with the strain of this that is most predominantly associated with toxic shock syndrome. And what you can see is that there are certain regions, different regions, that are going to be important for maintaining methicillin resistance, right? So it can tell us the regions of the genome that are really important. So, one thing that we can see is, like, for instance, R13, it seems like every organism needs to have this specific region of difference, this specific type of DNA in this segment, in order for there to be methicillin resistance, right? We can also see that there are other regions that seem to be pretty important and are commonly observed across these organisms. But this helps us get to identifying the regions of the genome that we then can associate with specific pathogenicity and specific resistance. And another thing we can do is we can look at something like toxic shock syndrome and show, oh yeah, look, we can see that all the organisms involved in toxic shock syndrome tend to have these regional differences at 3, 10, 13, 16, and 17. Like, these are important for it. So just some of the ways that we can start to think about how we might compare the genomes of these different organisms to better understand what are the components necessary for them to become pathogenic and also antibiotic resistant. So in all the examples that I've shown you so far, we've been looking at just comparing specific genomes, one genome to another genome or to maybe multiple genomes. But one of the things that's a challenge in microbial work is that what happens if we can't actually grow the bacteria we're interested in the lab? And this is not an uncommon outcome, right? Like, it's not every bacteria you can grow easily in the lab and harvest, nor will it maybe behave as you would expect it normally in the lab as you would in the environment. And so to address this, one of the big changes from a genomics perspective is by taking what's known as a metagenomic approach to studying microbial communities. And some refer to this as basically uncultured microbiology, which is a play on words, right? So it's kind of like uncultured because you're not culturing anything, but some might argue that it's uncultured because it's kind of coarse in its approach. But basically, what you do is you go out and you collect genetic material from the environment. And then you take that material and you can amplify it or you can sequence it directly, and then you're going to use those sequences to determine the species composition and abundance without culturing. So you'll be able to look and see what's in there without culturing, whereas culturing might bias you to what's actually available to grow in the lab. Now you can just see, well, what's there without me having to bias myself by lab work. And it's effectively a way that we can use genetics to look at community ecology. Traditionally, what has been done in this case is that we've used a common target, like a 16S rRNA gene that we talked about before in terms of showing those big differences between bacteria and archaea, right? But in this case, it's going to allow us, if we can amplify that gene, it'll allow us to amplify a target that we can find in any bacteria or archaea, right? And the advantage here is that this is a gene that's not prone to horizontal gene transfer. It's pretty well understood. There are some disadvantages, is that especially it doesn't actually give us much insight into what the organisms are doing. So yeah, I might be able to look and see if something is there and perhaps at how much of what percentage of the community it might make up, but I can't see what it's actually doing. And so to do that, we've now expanded things out to what is actually true sort of metagenomics, where we're actually assembling entire metagenomes or genomes from environmental data in order to identify functional components of genomes that are at high frequency. So it's almost like going back to that idea of, like, does it really matter so much that we have genomes of individual organisms, or is this sort of universal genome really the best measure of microbial communities when they're in actual nature and in the environment? This has spurred efforts like the Earth Microbiome Project, which is generating samples all over the world of different types of microbiomes: sediment, soil, water, animal secretions, intertidal, plant rhizosphere, et cetera, right? So there's all types of genomes that are now microbiome data that's being created in different parts of the world to try to catalog the various types of microbial communities that exist out there. To look at an example of how metagenomics and genomics approaches can be used to better understand biological systems, we could look at a system like marine cyanobacteria, and specifically the dominant two genera, Prokaryotes and Synococcus, and their distributions to better understand the biology of systems. So we can look at the basic processes of microevolution with regard to their application to this question, right? We know that organisms explore genomic variation. We know that adaptation will drive allele frequency change, and that neutral processes will also contribute to changes in allele frequency. So given that we can understand that these types of things are going to be occurring, we can begin to see if we can address some more complicated questions that we might be more interested in. What might be a little bit more challenging to understand in these cases are things like, how does environmental variation affect the splitting of populations or the different choices and strategies that are adopted? Do we see factors such as light and temperature, or a combination of factors affecting populations? Is this driven by genetics or protein divergence? Which proteins are the ones that are likely to be most important in supporting the evolution of microevolutionary change that we see in the ecological responses? How much novelty is needed to adapt, and where does it come from? And when we think about these different problems, right, we have to be able to disentangle a lot of different complicated relationships between the physical, ecological variables, the genetics, and the species biology in order to really understand these processes. And the ocean presents a nice system for this because we can identify a characteristic like depth that can sort of be a nice proxy for multiple different environmental factors. If we think about the two most frequently observed marine cyanobacteria species in the ocean environments, the two genera Prokaryotic and Synococcus, we find that they exhibit different levels of adaptation to light intensity. So we have strains of Prokaryotic that are adapted to high light environments in terms of the MED4 strain. It has a little bit smaller genome with fewer genes versus the low light adapted MIT 9313 strain. Also, we tend to see Synococcus in more of these low light environments as opposed to high light environments. And the data that we're looking here that's showing these abundancies, right, is the total percentage of sequences. So we're taking environmental DNA, we're looking at it, and we're seeing, how much sequence representing each of these strains do we find at each of these depths. And that's what's giving us this profile and allowing us to define where we would expect to find these things. As we examine the genomes for these organisms, we can see some of the differences and similarities between them. For instance, we can see, that the more light-adapted genome for MED4 seems to be smaller, has less genes in it. We can see that there's a lot of genes that are shared between all three species, so about a little more than half of their genes. We can see genes that are specific to the Prokaryococcus genus. But this pattern of gene loss, I think, is important, and it does show us some distinct differences where we can start to hone in and use that to identify, okay, what are some of the things that vary between these organisms that will allow us to explain their biological differences? What are some of the ways that genetics are similar and differ, right? This is the comparative framework that we want to use. What do we actually see in these systems is what's often accounting for the differences that we see in these in depth adaptations are genes related to processes involved in what's known as the phycobacillosome, or which is a photosystem 2 biological system that is found within cyanobacteria. And basically, Prokaryococcus uses PCB genes that can bind to modified chlorophylls to deal with various issues of depth and light availability. What we tend to find is that these genes are homologous with iron stress-induced proteins, and what they allow for is different amounts of chlorophyll, either A or B, to be expressed and utilized. And so what this allows is by changing the chlorophyll ratio for different types of chlorophyll that are better used for using high or low light conditions, we can see that each of these organisms has the ability to deal with different types of environments in a better way. So if we look at something like the low light adapted strain of Prokaryotes, it has more copies of these PCB genes, whereas the MED4 has protection actually against photochemical damage. So there they actually have phycoerythrin and photolysase genes that can basically help protect it against protect their genome against experiencing more damage from light. Another example of adaptive genetics in this case could be the differences in genes available for nitrate reduction. So here we see Prokaryotes losing some of the genes involved in this process, with the more highly light adapted ones becoming less able to process nitrate as compared to Synococcus. So all of this goes to show that when we look at these genomes, right, we can identify, what is the evolutionary process, sort of gene loss that might have led to sort of genetic change or differences between these two, as well as some of the specific adaptations, some of the specific either acquisitions or duplications or losses of genes that could be involved in biological processes that are useful to them. Lastly, I want to talk about viruses. Viruses are obviously very much in the news these days. If we think about viruses, we can think about what are viruses? They are basically, it's a nucleoprotein particle that has some sort of nucleic acid genome that's encapsulated by a protein or a glycoprotein coat or a viral capsid. And so it has a few very simple components to it. It's got the nucleic acid code in the middle, which can be either DNA or RNA. It's got a protein code around that nucleic acid genome. Then there's the envelope and the spike proteins that are all on the surface of it. The goal of a virus is to make copies of its genome and synthesize viral proteins, pack them up, and then get out of the cell, right? Very simple life cycle, very simple lifestyle, but it can be incredibly important to us both in our health and also the environment in terms of the ecology of disease. If you think about how much of the world's carbon is stored in the ocean, you think about what the role of bacteriophages could have on bacteria that are serving as that carbon source, you can start to see why they can actually have huge ecological effects as well. Think about the genome sizes of viruses. They tend to be between about 5,000 base pairs to as large as 2.5 million base pairs. And there are some sort of very specific types. So we have bacteriophages, which tend to target, we have single-stranded DNA viruses, of which bacteriophages are a good example of, which tend to target bacteria, right, as their host organism. There are double-stranded DNA viruses. These include adenovirus, smallpox virus, Epstein-Barr virus. We have single-stranded RNA viruses such as HIV, flu, and also coronavirus. And then there's also some double-stranded RNA viruses like blue tongue virus. Viral proteins, there's not a ton of different type of viral proteins. If we were to look at sort of the general categories of them, there would be about the 9 categories of genes that we see here. So, these refer to the GAG and the POL, the ENV, or the envelope protein gene, TAT, REV, NEF, VIF, VPR, and VPU. Each one of these proteins having a specific role in either in the viral life cycle. Think about what that viral life cycle is. It's pretty straightforward. Basically, you get a case where the spike protein and the envelope proteins interact with the CD4 receptors, something like the CD4 receptors on the surface of a cell. They attach, they then fuse with that cell. Their nucleocapsid covered nucleic genome is then exposed through an uncoding process. If it is something like a retrovirus, then a couple of transcripts will then be transcribed to produce a reverse transcriptase. That reverse transcriptase will then generate a suite of other proteins that will help import the viral proteins into the nucleus, especially the viral genome, and it'll actually invade and integrate into the host genome. Once it's in the host genome, it'll start to transcribe itself, just like any other gene, be exported from the nucleus to the translational machinery of the cell. It'll then create all the viral accessory proteins and all the major proteins that are needed for viral assembly and production, and then it will assemble the virus, which will bud off or explode out of the cell. There's kind of two different processes, and then be released out into the rest of the body. So that's the classic viral life cycle. Viruses themselves are used for all kinds of things. We use them in various ways in biotechnology. They're incredibly important. If we think about the various types of viruses, we can use them, things like adenovirus and other viruses, to do things like attack tumors and vaccinate an organism against the tumor, to do genetic manipulation and gene editing. So there's a bunch of different ways that we can actually use things like oncolytic viruses to have really positive effects on human health. So it's not just about the negative things that viruses. Viruses we've learned to use in a lot of ways to do really advantageous things for us as humans in our medical system. Viruses, some important things to know about the evolution of viruses is that they do evolve, and they're constantly changing, and they evolve at a rate which is much faster, almost 1 million times faster than other cellular life forms. In a large part, that's because they have such short generation times. Also, the machinery that's responsible for making copies of their DNA is also very prone to make mistakes, and they create tons of DNA, right? Tons of progeny. There's not a whole lot of junk in there, so every variant that you get out of there is often going to cause a real phenotypic change in the virus. There is virtually no error correction in DNA and RNA replicating viruses, so viruses with an RNA genome, and it basically means that error correction is really not a big thing for RNA viruses, which tend to cause a lot of the diseases that we see in humans. There are some things too where they can mutate too fast, and it can actually cause the virus to become unviable and unstable. It's kind of interesting how that works. So it actually mutates too much, and it ends up knocking itself out. So some examples of viruses. One of the classic examples is influenza. Every year, there's about 1 to 1.5 million people who die from influenza. Usually it's actually not influenza that gets them, but some sort of subsequent bacterial infection after their lungs are weakened. In the 1919 pandemic, influenza pandemic, there was about 50 to 100 million people who died from influenza, and a lot of them were actually young people. It's thought that these types of, if we were to have the same kind of influenza epidemic nowadays, it would be a lot worse because we have a lot of variable situations from what was happening in 1918, and I think the COVID pandemic is indicative of this, where people are able to migrate between countries and move between countries much easier and spread it. There are we have large older populations and other populations that can be somewhat immunocompromised. There's a lot of hypotheses about what led to the pandemic occurring. A lot of them are associated with the fact that this was at the end of World War II and soldiers were coming back from Europe, and many of them are bringing it home with them. Also, that there was just general overcrowding, poor nutrition, and healthcare for refugees. What was interesting is that it's thought that actually one of the reasons why the pandemic hit young people so hard is that they did not have immunity to this, because previous bouts of influenza that had moved through the population had actually left some of the elderly with more immunity, so they were actually able to handle this kind of thing. One of the key things, I think, in understanding influenza, because it is quite possible that we would run into another influenza epidemic in the future. We've had the COVID-19 epidemic that we're currently in. There's no reason to expect that we couldn't also experience one with influenza. In fact, a lot of people had predicted that influenza would be the next pandemic and not coronavirus. But they predicted something like it, right? So it could just be just as bad if it was to happen. And one thing to note is that influenza, what we tend to identify it is using two types of proteins to identify the strains or serotypes, and that's the hemagglutinin and the neuraminidase proteins, and they're basically like 13 and 9 types of these two different proteins respectively, and so we tend to define them based upon which type they have. So the 1818 to 1919 type of flu was called H1N1. So you might have heard these terms before when people talk about different types of flu virus. So typically the hemagglutinin is actually like a species-specific target. So like that sort of surface protein only works on certain organisms, certain species. So if we think about the case of something like avian flu, right? So this happens most of the time, but not always. Avian flu is an H5 virus, right? So the hemagglutinin on the surface is an H5, which is different than the H1, right? And so H5N1, right, versus H1N1, right? So the N1, the neuraminidase, is actually still the same, but the 5 is different. And so we think about, like, people always talk about, well, how is the next pandemic going to happen when it comes to this process where humans are going to be exposed to this, is that you could have something where you have a virus that's an H1N1 flu virus, an avian flu virus, infect domestic birds, right? And then they're going to be in close contact with something like pigs, right, which have their own HN target, and you could, but also pigs can be infected by strains that are that can infect humans. And so what could happen is you could have, this human strain and this bird strain intermixing within this intermediate pig host, leading to a completely new strain of flu that could go through and have incredibly dire effects, because nobody would have immunity to that flu. Like we haven't experienced a lot of H5 flu virus, right? And so if there was some way that these things can start to mix in these intermediate hosts, that becomes really problematic. And that's one of the main things that I don't think people always understand about viruses, is often it's the relationship and the proximity to some sort of repository a virus that is going to actually lead to the viral outbreak. Ebola is often seen as being another example of this, where Ebola, they think, was able to sort of jump from a primate species to human in some cases, right? And that may be to people sort of the practice of eating bushmeat, and that's one of the hypotheses around it. How did they get exposed to it? Well, this is a virus that ended up in primates, and then by getting close to them, that's what happened. And so, we've got a lot of things that make, and coronavirus has shown us this, that make us particularly vulnerable to these things. Increased population densities, long distance travel, bad public health policies. Not so much for coronavirus, but for flu, intensive livestock production is a major issue here there, especially in Asia, where these flu viruses tend to have these natural cycles of co-occurring. So that brings us to sort of our modern day case, right, which is the coronavirus. The coronavirus is also an RNA virus, a retrovirus, a single strand RNA virus. It's commonly found in lots of animal hosts, but these also often include bats. And the virus itself is actually the severe acute respiratory syndrome coronavirus 2. That's led to what we refer to now as COVID-19, which is the actual respiratory disease. Its genome is about 30,000 base pairs long, a little bit less than that. It's got about 14 open reading frames. And this isn't the first time the pandemic that we're experiencing right now isn't the first time that coronavirus has affected. We've seen evidence of infecting people. We've seen an outbreak of SARS in Asia in 2002, 2004, and another outbreak of coronavirus in the Middle East, where we had about almost 600 people die in about 26 countries in 2012. But you'll notice that, those outbreaks were much less severe than what we see in coronavirus, and that's because this version of coronavirus has a lot of adaptations that allow it to be much more transmissible. Its replication process is very similar to your average viral process. Basically, it attaches, it fuses with a cell, and then starts replication. Initially, it'll just produce two polyproteins, and then these two polyproteins will then interact and generate this replicase polymerase and generate a whole bunch of smaller proteins. It'll then work its way into the endodeposited reticulum and then start creating coronavirus proteins, which will then create coronavirus particles, which are assembled and exported from the cell. So as you know, we've been in a global pandemic with coronavirus, SARS-CoV-2, and COVID-19 since January, February of last year. So really sort of ramping up in March, right? Our own university closed down, everything shut down. We look at how it's looked for the entire world during that time. We've basically seen, a big increase in cases, or a steady increase over time for a while, and then sort of big bumps, and we're now sort of in our third big bump currently. There's been over 223 million, almost 224 million cases reported up into this date, and that's probably an undercount of how many cases, because not everybody can get tested and identified as having coronavirus, especially worldwide, where tests aren't readily available in all countries. There's been at least 4.6 million deaths, if not more. We think about, remember I showed the flu epidemic, right? There was about 1 to 1.5 million deaths. So, pretty dramatic experience. Obviously, a lot more people in the world now than back then. We can see that, the pandemic has affected different regions more than other regions, specifically North America and the U.S. It's been particularly bad for us. It's also been bad in the Middle East and Europe and other places. And again, this is all dependent upon people reporting well. So, there's some reasons to expect that some places are not reporting as well as other places. And this is data all taken from the New York Times just the other day. What we can see in terms of like what are the hotspots just currently for coronavirus right now, we can see the U.S. is still one of the big ones. Parts of Europe, parts of Asia, Indonesia, and we see other red parts coming up in various parts of the world. So a lot of concern. It's still ongoing. We can see that if we just look at the U.S., there's been about almost 41 million reported cases at this point, accounting for what is an estimate of over 65,000 deaths. This accounts for of the total number of identified deaths for COVID-19. This would be about 14.25% of the total deaths for coronavirus that happened in the U.S. and about 18% of the cases. At one point, it was as high, I think it's like 25% of the cases in the world. Considering that we account for only about 4% of the world's population, it's disproportionately affecting our country more than others. If you wanted to think about how it's affecting us more locally, we could look at California, and this is the data for California. We can see those same three spikes that we see in these other ones, right? We saw these spikes that we saw back here, 1, 2, and 3, so November, sort of May, August, right? We can see that these spikes are, the one in November really affected the US and Canada a lot more, Europe than maybe some other places in the world, whereas the one in the summer had a larger effect perhaps in Latin America and the Middle East, and this current one is really affecting us. And you can see that in the data where, these recent spikes are having a larger effect on our population than others, than the other ones. We can see, again, in California, we mostly have had two of these bumps where we had that winter bump and then the recent one here. Specifically, in Monterey County, right, our data looks a little bit like this. You can see that we are now weathering the storm pretty well with regard to the most recent bump. But there's been about almost 50,000 cases just in Monterey County during this time. I think one of the interesting things to think about from a genomics perspective is how this process has affected how genomics has been involved in helping this process. So in one, we can look at genome sequencing now has become commonplace all over the world, and people are actually sequencing the genomes of coronavirus in order to track the different strains, because these strains have been associated with different types of peaks of this virus. We know that the viruses that we talked about evolve very rapidly. We know that they're changing rapidly. By tracking these strains, we're best able to identify, what are the patterns of evolution that are occurring in which strains, and what changes are we seeing biologically in the organism that may be changing how the virus is behaving and how that might affect what we need to do from a public health standpoint. So through genome sequencing, right, we've been able to track the advent of these viruses. And so this is showing the different strains up here. These identified as clades. This is a tool called NextStrain, where it's showing us the different strains that are occurring through time. Right now, we're back in April 2000, right, 2020, excuse me. And we can start to see coronavirus popping up all over the world. And we can see the various strains popping up. The pie charts are indicative of kind of how many cases we're seeing in different places. Then we can see different strains like the alpha strain, the epsilon strain, which was many people associated, also known as the UK strain, and started spreading in various places all over the world, having a big influence, right? And we can see that it's actually different strains that may or may not be more transmissible taking over in different parts of the world. And we see these colors start to go up and down. The alpha strain starts popping up. We see the lambda strain coming out of South America. We see the mu strain, which is currently of major concern because people think it's resistant to vaccination. And then we see now the delta strain coming on and coming strong. And what we're seeing right now, right? So this is sort of the spring jump, right? We saw that big bump in the spring, and now we see this big bump that's blowing up in August right now all over the world. And this is basically the delta strain taking over almost everything. So the delta strain is really sort of dominating the landscape because we've seen a major change in the biology of this organism. So through time, we see all these mutations occurring, and we see this organism becoming more and more virulent, more and more transmissible, more and more prevalent, and easily passable. We also see it becoming more able to break through things like vaccination. And so this is just one of the things that happens through time, but it's only through genomics that we can actually able to see this. If before we had genomics, it would have been very difficult to identify these differences in strains and have an understanding that maybe we should be doing things a little bit differently if Delta variant has a different biology than what we were looking at back at the beginning of the pandemic. Right? So we can see also this tree represents all the evolution that has happened during that time, and we can see how Delta variant has moved away and is a very different organism at this point. So these are some of the ways that we could think about, the why it's so important to have these genomic tools at our dispense. Another thing that's really important that genomics is a huge event is in the actual production and our ability to make vaccines. So as many of you know, the vaccines that are dominating the landscape and that have been proved most effective, we've developed vaccines for coronavirus at a rate that's much faster than any other organism. Our first genome sequence for coronavirus, I think, was in February, which is just after it had come out. Right? And so because of that, folks have been able to identify the parts of the virus, the spike proteins, for instance, in this case, that are going to be the best thing to target for a vaccine. And they've been able to develop these mRNA vaccines where they can actually get the sequence that codes for a piece of the viral spike protein, not the whole thing, just a piece of it. One that is not going to be prone to, that they think is not going to be prone to mutation, so that it's going to be relatively stable and be targetable across multiple strains. And then they've basically been able to, with the technology we have now, they can actually produce this mRNA not in vitro. They don't have to do it in vivo anymore, and put it into some lipid coated structures. Right, kind of like a virus, right, and then inject it into cells. And what these mRNAs do is that they then interact with our own cells and get our own cells to actually produce little tiny spike proteins that are displayed on the surface of the cell. These spike proteins are then recognized by antibodies in our own body, and those antibodies then trigger our immune system produce more antibodies that will fight that spike protein. And this is, sort of the key. Like this part is very similar to what we would expect from a traditional vaccine, about how vaccines work. But the process by which we're able to generate this target, right, could only have been done through the advent of genomics and the application of genomics. So it's been incredibly useful. It's been incredibly important in this process. And as we see, like, what is the effect of vaccination being? This is kind of a color map. Darker colors indicate more vaccination versus less vaccination in the U.S. right now. Again, this is taken from the New York Times today. And what we can see is that if we compare it to sort of the hotspots, we see Delta variant sort of blowing up all over the place. And while we know that now the vaccines are a lot less effective at keeping people from catching coronavirus, they're going to be a lot more prone to being able to have a breakthrough case because of the capacity of Delta variant to break through vaccination. One thing I did want to point out, if you were curious, Monterey County ourselves, we're about 67% vaccinated for plus people who are 12 years and older. That accounts for about 56% of all the residents. Oh, my sister lives in Montgomery County. They're about 87% vaccinated for individuals who are 12 and older, and that accounts for about 70% of their total residents. So there are some, still big differences in these shades of green. And if we think about like what's the benefit of vaccination, right, we still see high rates of hotspots of coronavirus cases across the country. But if we look at patterns of hospitalization, one of the things I think that's really telling is that if we compare the regions where we see high vaccination rates, say the Northeast, say various parts of California as opposed to the Valley, right? We can look back and we can see, in these areas where there's less vaccination, we're seeing much higher rates of hospitalization in those areas. So we are still seeing coronavirus cases in lots of different areas. We do see, I think, reduced cases in areas that are vaccinated as well. But still, we can see it. Florida, for instance, we still see a high rate of cases, even though there's a lot of vaccination there. It's probably due to a lot of the fact that the older population is heavily vaccinated in Florida, whereas the younger population is not, and they may be more responsible for those outcomes. But what we are seeing is that where there is high rates of vaccination, we do see a decrease in the hospitalization rate. I think a really telling one, for instance, like if we look like this down here in Texas, right? High rates of vaccination in Texas, low rates in other parts of Texas. And if we compare the rates of the level of hospitalization, we do see what I think is like slightly better outcomes along some of these more vaccinated parts of the state than in other parts of the state. So just some interesting observations in terms of how this goes. But I think the lesson that I want to take you guys to take from this is that we would not be in the place we are today without genomics and comparative genomics. So it's really been a huge part of our ability to respond to this. And so thinking about why it's important to study and understand the genomes of prokaryotes, there is no better, I think, indicator of that than this example of coronavirus. So thanks for your attention.